Hey, it's 12 noon, so we'll get started. Thanks everyone for joining us, either in person or online. For those of you who are at the Glen, you're welcome to join us in person at D02-1312. We have our speaker and a light lunch here, so you're welcome to come in person. Thanks very much for coming. A brief land acknowledgement before we begin. McGill University is on land which has long served as a site of meeting and exchange amongst Indigenous peoples. We acknowledge and thank the diverse Indigenous peoples whose presence marks this territory on which we now gather. Just a reminder to please enter your questions in the chat at the end of the presentation, and please answer a brief survey that will be sent to you immediately after the presentation. It will let us know that you are present here with us today. And finally, our speaker is courtesy of the Division of Hematology, and I'd like to ask the Chief of the Division of the Hematology, Dr. Chantal Séguet, to introduce our speaker, please. Thank you.
Thank you.
Thank you very much, Dr. Janetti. First, I would like to thank Dr. Grignano for him to be present today to give us a talk at the medical grand rounds. Dr. Grignano was just recruited under the Division of Hematology and started last week. And without any hesitation, he accepted to give the medical grand rounds today. So I'm very, very happy that he has accepted, and I thank him for that. Just to tell you a little bit about Dr. Grignano. So Dr. Grignano is a physician specialized in adult clinical hematology with over 10 years of combined clinical and research experience. After a 6-year residency across major Paris hospitals, including a research year at the Gustave Roussy Institute, he served as a clinical fellow in hematology at the Cochin Hospital in Paris from 2017 to 2019. He subsequently pursued a PhD degree from 2019 to 2022, focusing on the ferroptosis in acute myeloid leukemia, while maintaining part-time clinical practice and teaching medical students. His postdoctoral work at the Princess Margaret Cancer Centre in Toronto explored mitochondrial metabolism and inflammation in TET2 mutated clonal hematopoiesis. He is now beginning, as I mentioned, an appointment as a clinician-scientist here at the McGill University Health Centre, where he aims to advance research on iron and mitochondrial metabolism in acute myeloid leukemia. So today, Dr. Grignano will talk to us about mitochondrial DNA mutations, contribution in hematologic malignancies. Is it simply incidental? So Dr. Grignano, we are very excited to listen to your talk today. Thank you.
Thank you, Dr. Séguin, for the kind introduction. And thank you all to be here, online or present. So yes, today I will talk about mitochondrial DNA mutation contribution, as Dr. Séguin mentioned, in hematological malignancies. Is it simply incidental, or is it something that we have to take into account? And I'm asking the question also to my colleagues in hematology, maybe to draw attention on that specific point. So I have no conflict of interest yet. So just a brief introduction for those who are not familiar with mitochondrial DNA. So mitochondrial DNA, it's inherited from the mother. It's derived from the genome of bacteria that had been engulfed in the modern eukaryotic cells. It's a quite simple DNA, let's say. It's a circular one with 16 kilobase, which encodes for 13 proteins, most of them coding for mitochondrial proteins that will be part of the electron transport chain, the complex 1, 3, 4, and the 5 one, which is the ATP syntase, and also 22 transfer RNA and 2 ribosomal RNA. I think one of the most fascinating things about mitochondrial DNA is the high rate of mutation, supposedly 10 to 17-fold higher than in nuclear DNA. And one of the reasons for that is because mitochondria lacks most of the DNA reparation mechanisms like HDR or NHEG. And so that's one of the reasons. There are another reason, but I won't extend too much on that part. Mitochondria is, as you know, mostly involved in energy production, oxphos respiration, and it accounts for up to 90% of the cellular energy in the cells. And it has all the function, obviously, intracellular signaling, apoptosis, and so on. So I think one concept to apprehend is the heteroplasmic concept. So basically, you have several mitochondria per cell and several mitochondrial DNA copies per mitochondria. So if we sum it, you can have up to 1,000 of mitochondrial DNA copies per cell. And when you have a mutation on one of the strands of the mitochondrial DNA, it can be 0.5, 0. something percent up to 50, 60%. So it's kind of a variant allele frequency type of thing, but it's very specific for mitochondria. So when I mention heteroplasmy, it refers to the mutation load of the mitochondrial DNA in the cell. So what scientists agree on is that to have a specific phenotype that can be seen in patients, you have to get over 50% of mitochondrial DNA mutation. Below that threshold, it supposedly can be like silent phenotype. So I'd like to just tell you a brief story that caught my attention when I was preparing my slides on the power of heteroplasmy mitochondrial DNA assessment. I guess you all know the Romanov family, the last Tsar of Russia. So the whole family has been slaughtered, killed after the Soviet uprising in 1917, and their bodies were hidden and buried for more than 80 years until 1991, where under Boris Yeltsin and after the collapse of the Soviet regime, they wanted to get the bodies and authentify the whole family. So overall, they found 9 skeletons out of the grave, one being presumably the Tsar, his wife, daughters, and some relatives. And so when they assessed the mitochondrial DNA, it turned out to be a mismatch between the Russian Tsar DNA and some of the living relatives back at the time. So it was kind of problematic for the regime because it was like fueling concerns about the authenticity of the remains. So they tried to postpone the authentication. They asked several teams all around the world. And one of the teams that was based in the US, they again compared the mitochondrial DNA, so the heteroplasmic concept back at the time, and they compared it to one of the brothers of the Tsar, which is on the right side, the Grand Duke George Alexandrovich, which died 20 years earlier, at the late 19th. And they found out the same heteroplasmy on the brother, in the brother. And so that's how they authenticated that the body of the Tsar was indeed the body of the Tsar. So I think it's a very interesting story because it showcases how a mutation in the DNA, mitochondrial DNA, can be very useful. So there is other application, of course, of the mitochondrial DNA heteroplasmy assessment. So lineage tracing, as I just mentioned, but also clonal inference or clonal architecture, in vivo dynamics. Some people even used it to do TCR cloning. In most of the case, when they compare the assessment of the mitochondrial DNA and compare it to the state-of-the-art techniques like single-cell ATAC-seq or RNA-seq, it turns out to be as efficient as the other techniques, sometimes more efficient. And for my colleagues in hematology, now they're using it also in hematological malignancies to use it like a kind of barcodes and to replace the single-cell RNA-seq. So which kind of diseases are associated with mitochondrial DNA mutation? Hopefully, rare disease. Most of them being neurologic diseases. So I don't know any of them. I just them like this, but I think the most frequent one is the Miller syndrome. It's 1 people out of 4,000 to 5,000 people. It stands for mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes. Maybe for the neurologist, it will be something familiar. And the Leigh syndrome is at the bottom part. I just saw a funny thing. It's funny, it's not funny, but it seems to be more frequent in some Quebecois population in Saguenay. And it's called the French-Canadian mitochondrial disease. I just found it out before starting my presentation. So why would we study the mitochondrial DNA mutation in hematological diseases? So when I was in Toronto doing my postdoc, my previous PI, Stephen Chan, he did his postdoc in Stanford, and they sequenced more than 100 people with AML. And what was striking is that they found out that 40% of people had mitochondrial DNA mutation in the electron transport chain gene. It was the second most frequent mutation after FLT3, which is the most mutated gene. And it was even more frequent than NPM1. So for those who are not hematologists, it's a nucleophosphine 1, which is one of the most frequent mutated gene in the AML. And so they tried to correlate it to other known mutation in AML. There was no significant correlation. So what we've done, we did the same thing by reanalyzing the TCGA dataset, the cohort of AML patients. So in the TCGA, you have more than 200, but just 200 AML patients with mitochondrial DNA sequence. So in this cohort, the mutation was less frequent. It's here, so it was roughly 8 to 9%, if I remember correctly. And when we did the same thing, trying to correlate the mitochondrial DNA mutation presence with all the known mutation, in that cohort, it turned out to be frequently associated with TP53 mutation. So it's few cases, but still it was significantly associated. And there was a trend for less co-mutation of NPM1 and FLT3 in that cohort. So when we pulled together all the data, we saw a significant less association with NPM1 mutation, and also a trend for FLT3 less associated with mitochondrial DNA mutation. We, of course, tried to see if there is an impact on the prognosis in those patients. So in the Stanford dataset of my previous PI, there were at least clear significant worse prognosis impact of those mutations in the mitochondrial DNA. In the TCGA dataset, it was the same, but keep in mind that it was associated with TP53 mutation. So maybe TP53 mutation accounts for the worst prognosis associated with mitochondrial DNA mutation. But still, there is a trend for those mutations to be of worse prognosis. So 2 years ago, a team took the data of the UK Biobank. So as you know, UK Biobank, it's a huge, large data set of people from the 2000 and onward years with material for hematological malignancies and other malignancies. And they tried to see in this cohort how many patients had single nucleotide variants in the mitochondrial DNA. So it turns out to be roughly 30% of the patients. They took a VAF above 5% as a threshold of positivity. And they correlated with the hazard ratio of any cause of death. So as you see here, I don't know if you see my pointer. Yeah. There was a significant trend for higher likelihood of death with a hazard ratio of 1.3. And when you look at the different type of cause of death, you see that neoplasm was significantly associated with the higher likelihood of death. And when they broke down the type of neoplasms that were significantly associated with a higher cause of death, it seems that hematological malignancies were the most prominent one. And among them, leukemia with a hazard ratio of 5, which is very significant for this large cohort. Then in the same study, they pulled out the specific diagnosis associated with the presence of mitochondrial DNA mutation. And if you look here in the red circle, you see that most of the cause were of hematological origin. So most of them were neoplasm, like acute myeloid leukemia, myeloproliferative disease, some lymphoid neoplasms, and also, interestingly, some benign condition like neutropenia, leukopenia. So it was pulled out from the patient record, but still it's caught my attention because it's significantly associated. And when they tried to break down the type of mutations that was specifically associated with the hematological malignancies, the one that stands out is the complex 1 mutation. So complex 1 is the first complex of the electron transport chain. It's the most important, let's say, in terms of functionality. And this one was significantly associated with all sorts of hematological conditions. So why mitochondrial DNA mutation would be selected in specific clones? So HSC stands for hematopoietic stem cells. So why those mutations would be retained in the context of hematological malignancies? So, well, there is 4 types of phenotypes associated with mitochondrial DNA mutation. The first one is less interesting for us because it's the one associated with apoptosis. So basically, a cell acquires a mutation. It will lead to a cell death. It's the kind of phenotype that we see in neurological diseases. The second one would probably interest the oncologists because some papers stated that upon the acquisition of mitochondrial DNA mutation, you have a Warburg effect, so an increase of the glycolysis shift, and this will in turn lead to a progression on some type of cancer like colon or kidney. And for us in hematology, I guess the 2 associated phenotypes out of the literature are the one affecting the OXPHOS capacity. So there is a high literature on how in specific hematological malignancies you have a shift of the OXPHOS capacity of the cells, and the other one being the inflammation. I will touch on that later. So those 2 specific phenotypes associated with mitochondrial DNA mutation are the ones that may explain and drive the expression of the clonal disorders. So I put it the slide. So for my colleagues in hematology, they won't learn so much on that slide, but for the others, maybe it can be interesting. So how hematological disorder arise? So first, you have the appearance of what we call a clonal hematopoiesis associated mutation. So there is 3 main mutations concerning epigenetic regulators here on the left part, DNMT3, TET2, and A6L1. Most of them are epigenetic regulators. Sometimes it's signaling molecules like JAK2. Later on the disease, you will have the acquisition of other mutations, either affecting the splicing factors or the DNA damage response. And later on, and this part is the one that most of the scientists try to study and to break down, is which kind of specific selective pressure can be applied on those early clones to drive and to turn them into overt hematological malignancy. So the one being studied is inflammation because there is a lot of literature supporting this type of selection pressure, but you can have also microbiota, and some people are studying this aspect. But whatever the pressure would be, which is applied on the clones, it can sometimes turn into a myeloid malignancy, so a myelodysplastic syndrome, and later on, upon the acquisition of another mutation, will turn into overt AML. So as I mentioned, the inflammation in one of the most studied selective pressure on clonal disorders. So there is a very huge literature on that topic. So I just picked some of the papers that I think are the more interesting. To put it in shorter terms, many people, so we decided to study the TET2 mutation, one of the mutations affecting clonal hematopoiesis. So it's the second most frequent mutation found in patients. It can be like over 10% of patients above the age of 60 that have CHIP mutations, and among them TET2. But then there is a lot of papers stating that inflammation is very important for the clonal expansion of TET2 mutated clones. So most of the inflammation pathways have been studied. IL-1 pathway, IL-6, and STING. STING, I think, is the most relevant one because we know that upon mitochondrial DNA mutation, mitochondrial DNA leakage in the cytosol, STING is one of the most activated pathway inflammation. And in all cases, people have shown that when you apply a pressure with inflammation, whatever it is, you will drive the expansion of a CHIP mutated clone. So I just, as an example, put this paper. So basically, if you take a mouse, if you delete TET2 in one of the allele, and if you treat the mice with LPS, so liposaccharides, after 8 months, the TET2 mutated clone here in red will take the lead and will overgrow compared to the other clones. So this is true for many types of inflammation. And when you look at the spleen of the mice, it's tend to be increased in terms of size. In the mouse, the TET2-associated phenotype is more likely a myeloid myeloproliferative neoplasm, which is quite different in human, but still. So I'll just take a step aside on my presentation. It's not my main topic, but I will just touch on that because I will later describe what we've done in my previous postdoc. So a few slides about gene therapy. So I guess many of you are familiar with gene therapy techniques. There are 3 types of gene therapy that has been used by scientists until now. The zinc finger protein gene therapy, the TALENT gene therapy. So TALENT stands for, I don't remember, but I know it's closed. So TALENT is a kind of technique which uses amino acids that will recognize a DNA sequence. In this technique and the other one, it's associated with an endonuclease, which is FOCL, which will induce a double-strand DNA break. And this will lead to a mutation in the DNA that has been targeted. And the last one, as I guess you all know, CRISPR technique, which has been used. I guess it's the most used technique in the last 10 years since it was discovered by Emmanuel Charpentier and Jennifer Doudna, in which you have a target DNA sequence which will assemble with the Cas9 nuclease and which will lead to a double-strand DNA break. So those are all the techniques that we use to induce a DNA mutation, and it's also true in the mitochondria. And I guess the gene therapy field took a milestone a few years ago when a new gene therapy were FDA approved in sickle cell disease. So this has shed light on the gene therapy techniques, not just only for bench perspective, but also applicable to patients. The last one, maybe just a slide on it, because I guess we will hear more and more about it in the next few years. It's a prime editing technique. So prime editing, it's maybe the perfect technique from many points of view because it allows you to induce a point mutation in whatever DNA sequence you want by giving to the cells a nickase, which will just cut one strand of the DNA, and you bring a template, a donor sequence, and you are basically theoretically able to generate whatever mutation you want in whatever sequence you want. And so this technique has been applied successfully by this team in Boston in 2023 in sickle cell. So as they applied the prime editing technique in hematopoietic stem cells in sickle cell disease patients, and they saw that with the gene therapy technique, they were able to reverse partially the phenotype of the sickle cell. So you see here, for those who know how to read a smear, you have less sickle cells after the gene therapy has been applied. This is a mouse model. So to get back to my presentation, so the question was, how can we generate a DNA mutation in the mitochondria? Which technique can we apply? So we tried to focus on the technique that I mentioned, the Talon one, which is a sequence of amino acids that each sequence recognizes a DNA. They have base pair. It's linked to deaminase. So this deaminase will turn cytidine into a thymidine. So you are able to generate a point mutation in the specific sequence. And so we decided to target the mitochondrial ND5, which is part of the complex 1, because as I told you before, complex 1 is one of the most mutated gene, and it's very important for the cell. So we generated 3 types of mutation. I won't extend too much on that part, but a synonymous mutation, a missense mutation, and a nonsense mutation leading to a truncated protein. The 3 of them has been described in patients' context. So again, it's another slide to picture you how it's working. So you have 2 arms that will hybridize, specifically recognizing the DNA sequence. And upon the hybridization, you will have the deaminase that will reconstitute and induce a point mutation in the DNA. And so, of course, you can sequence the DNA to verify that the mutation has been put in place. To conclude my topic, so I presented the data on AML, acute myeloid leukemia. But I guess the most relevant question is to assess those mitochondrial DNA mutations in the context of CHIP, so clonal hematopoiesis, because we know that if we can intervene early in the course of the disease, maybe we can prevent the appearance of a more overt disease. So the same team that published the previous paper I showed you issued a paper last year. So this time they tried to look at the presence of heteroplasmy in a cohort of patients with clonal hematopoiesis. And what they find is interesting because when they compared the 2 populations, so people with clonal hematopoiesis and people who don't have clonal hematopoiesis, it turns out that the heteroplasmy, so meaning the mutation load in the patient with CHIP was higher. So it was true in the UK Biobank cohort. It was always true in another cohort of people with cardiovascular disease. And they try to depict a little bit specifically in terms of mutation they found in those patients. And the 2 things that caught my attention, first of all, patients with a higher mutation burden, CHIP burden, they displayed a higher mutation load in the mitochondria first. And the mutations that were significantly associated with a higher mutation load in the mitochondria were the mutation affecting the spliceosome. So overall, it seems that those 2 patterns, so the mitochondrial DNA mutation and the mutation affecting the clonal hematopoiesis, seems to work closely together in some context, specific context, to drive the hematological malignancies. And this can be seen in this cumulative incidence curve. So again, it's a very high amount of patients, so I think it's very significant. If you take the patient with both clonal hematopoiesis and mitochondrial DNA mutation, those patients seem to have a very high likelihood of developing myeloid malignancies, hematological malignancies. And it was 12 times a high likelihood compared to naive patients, and it was twice more than CHIP-only patients. So I think it's very important to take that into account. This is my last slide, my key messages. So first, mitochondrial DNA mutation are commonly seen in people with hematological malignancies, and it seems to confer worse prognosis. It affects the host force capacity of the cells, as well as inflammation in those cells, and it seems to lead to a more aggressive phenotype in mice. And the mitochondrial DNA mutation seems to be very prevalent in patients, especially in patients with CHIP, and seems to drive a higher likelihood of progression to an overt disease. So just to take a step back, why it is important, CHIP affecting a lot of patients above the age of 60. It's more than 10% after the age of 16, even sometimes 20% at the age of 70. The drivers that lead to the clonal expansion in CHIP, in clonal hematopoiesis, are not well understood. There is now a lot of supporting data about inflammation, but what drives the inflammation is not well known currently. So I think mitochondrial DNA mutation is not the only explanation, but can be one of them that could explain in part the phenotype we see. And even though there is no applicable therapy, I'm just anticipating the question on mitochondrial DNA mutation. I think still there is maybe something we have to take into account and to assess in patients, because it can allow us to better predict the likelihood of progression in those patients, especially the patient with TET2, DNA mutation, and spliceosome mutation. And that's my last message.
Super. Thank you so much. Thank you. Very interesting, comprehensive lecture on this very important topic. Are there any questions in the audience, or you can enter your questions in the chat? Yes.
Hi, Eric, great talk. Just a couple of questions for you. One sort of more broadly applicable to the audience, and one a little bit more esoteric. If DNMT3A and TET2, I mean, they're both CHIP mutations, they're both involved with abnormalities of methylation, hypermethylation. But only TET2 is really associated with accelerated progression and more oncogenesis. Why not DNMT3A as well?
You mean in the context of mitochondrial mutation, or generally speaking?
In the context of your mitochondrial mutated, yeah.
So no, DNMT3 is also associated. So we decided to focus on TET2 because the inflammation, which is a phenotype associated, one of the phenotype associated with mitochondrial DNA mutation, is specifically studied in TET2 context, less in DNMT3. Even my previous lab published a Nature on DNMT3 and metformin usage to prevent the inflammation. But TET2 is the one that I think is the more relevant one, I guess, for us. Now we know that spliceosome mutation may be most relevant, even more, but it just issued at the end of my postdoc. So,
and then the other question, any association between, this inflammation of mitochondrial mutation and the phenomenon that seems in CHIP, it's not just myeloid malignancy we worry about endovascular damage. So
yeah, of course.
Rates of heart attack and CVA are higher in people who have CHIP. Is this?
Yeah, I think this is something we have to take into account. So maybe we have to talk to the cardiologist about this topic. But yeah. Oh yeah, sorry.
We'll talk after. Sorry. Yes.
So I think you talked most about lymphoma, leukemia, but I was wondering, in lymphoma, is this also important?
So in the UK Biobank cohort, it seems to be also associated with some kind of lymphoid disorders like the CLL, so chronic lymphocytic leukemia. But it was less prevalent, and the prognosis impact doesn't seem to be associated in lymphoid disorders. Even though TET2 is mutated in some kind of lymphoid disorders, we know it can also affect lymphocytes, but we didn't focus on lymphoid disorders for this project.
George?
Yeah, nice question. Are all the mitochondrial mutations associated with increased activity of the Krebs cycle and electron transport chain and increased ROS?
So the one we studied in complex 1, yes. It was surprising for us because when we generate the mutation in the complex 1, we expected a lesser activity in terms of OXPHOS capacity, but it wasn't the case. So I didn't show all the data. We saw that some other mitochondrial function were affected, like mitophagy, mitofission, and that maybe explain why they have a higher OXPHOS capacity. So I can't speak for the other mutation, but for that one, clearly it was the case, what we studied.
So just in follow-up, so there are agents that can decrease oxidative phosphorylation.
Yeah.
So have any of them been used, like maybe metformin inhibits complex 1.
Yeah, we tried metformin, it's working. It's working. So, yeah, I think among all the inhibitors that are available currently, metformin is the one that catches the attention of everybody because it's cheap. We can use it in patients. It's quite safe. And it's so in a Nature paper that we issued last year, we saw a very drastic effect in the NMT3 context in mice. So you have to be an patient, of course. But there is big, large cohort data. I guess the Swedish published the data. They looked at all the people with CHIP, and they just picked out the patient with taking metformin, and it seems to be also an effect in patients. So less hematological disorders. So I think there is more work to be done, but it's interesting.
So maybe on that note, I just wanted to ask if you have to treat or you think you would treat them when they have CHIP before the malignancy, or will it still work in progression of the malignancy?
I can't tell. I don't have the answer of the question. But the more early, the better, I guess.
I'd like to go to the questions in the chat. There's a couple of questions in the chat. Would you expect cells with mutations in mitochondrial DNA affecting complex 1 to be more or less dependent on
Vaginophagy.
Yeah.
I'm smiling because it's my other topic of my heart, vaginophagy. I don't know, actually. I don't know. But that's one question I want to assess. I don't know who's has the question. Yeah, okay. I see.
And there's a question if you can see a mitochondrial DNA variants known being pathogenic or not, all are well studied.
So what we know is that the less frequent mutations are the ones that the more functionally impact the cell. So let's say that the mutation affecting complex 1 are less frequent than other mutations, but that's the mutation that affects the most the cell. If you take a mutation in the transfer RNA or ribosomal RNA, it won't be as impactful as the complex 1 or complex 2. So in the hematology context, the patients that have mutation is mostly complex 1, complex 3, complex 4, and less of the other mutation.
Just a few words about, so welcome to the MUHC. You have a recent position, I think a week ago, so congratulations. You know, you have a broad medical audience here. What are you going to be doing at the MUHC? Do you want help from your colleagues from other specialties? Are you looking for databases? Can you give us an idea of what your plan is?
So thank you for asking. Yeah, so my plan is to continue my work on mitochondrial metabolism, and iron also is one of my topics that I like the most. So for now, I'm doing mostly in vitro bench experiments, but I think later one question that needs to be answered is in the CHIP patients, so clonal hematopoiesis patients. I know that there are some ongoing experiments. I went to Rosemont, and one of the team is studying with cardiologists to assess the effect of colchicine in patients with CHIP. So I think it will be useful to see on a large cohort of patients affected by clonal hematopoiesis how early intervention can reverse the phenotype. And so colchicine may be one example, but I think there is also an example, metformin being one of them. So I think there's works to be done, and maybe that will be my next move.
You screen for patients with CHIP, and what is that a costly thing to do?
Mitochondrial DNA, you mean?
Yeah.
It's not costly. I mean, it's not more costly than assessing DNA mutation. So if you do a whole DNA sequencing, you can catch the mitochondrial DNA. So I guess you have to focus on it. You have to look at it. So people often overlook mitochondrial DNA, but I think we have to focus more on it.
So in that line, what would be your population that you would use to study the...
You mean for a clinical study perspective?
Yes.
I think the people in which a mutation has been found. So I know that we are not assessing patients because most of the time they don't have any symptoms. Mm-hmm. But let's say if you take the sickest patient, so sickest, it's people with a mutation and a cytopenia, and you found, let's say, a TET2 mutation, I think those patients are the ones you will catch and the ones that you can try an early intervention to see what's happening.
Dr. Rosenblatt, you have a question. I'm wondering if you want to unmute yourself, David, and ask your question. We have a few minutes. Go ahead.
Yeah. I think Dr. Buhas also had a question about were the DNA variants mentioned known pathogenic, but also there are many probably cell lines available from patients with the primary mitochondrial diseases, and I don't know if you know Eric Schubridge at the Neuro, who's a world expert on these, and Daniela Buhas has a clinic following them as well clinically at the MUHC. But I was just wondering whether any of these cell lines, fibroblast cell lines from these, are useful in looking at any of the models that you're studying or any of the mechanisms you're studying. Maybe fibroblasts are not a good model.
Maybe I can't tell. I didn't used to work with fibroblasts, but I think it can be assessed. I think the microenvironment overall, it's not so well known in hematological malignancies, and maybe mitochondrial mutation can be also affecting microenvironment cells, whichever fibroblast or other cells. But I can't tell. I didn't study specific question.
Thank you, David. Gail Breton, did you want to unmute yourself to ask your question? Otherwise, the question is about the mutations in metabolism, dysregulation, acute or long-term COVID.
I'm not an expert at all on that specific topic. I can't tell, but maybe you have some clues into COVID. I don't know. Maybe.
So unanswered questions. Are there further questions? Oh yes, George.
Question. So you mentioned the loss, the reactive loss of the species that shows the variants. What do you think about antioxidants and the inflammation? Is that clearly related? Is it due to the increased ROS?
Yes, yes, there is a. So when you have more ROS, in most of the case, you have inflammation because ROS is one of the driving cause of inflammation. So in the IL-1 pathway, it is well known that ROS at some point will trigger the IL-1 beta production. So it's known, and if you buffer the ROS, it's obvious that you will turn down the inflammation. That's why we use it. So I think just to extend your question, there is an ongoing trial that is interesting. They use anti-IL-1 antibody that already been used in rheumatology in the context of, I think it was MDS, but I have to double check. So I think it's one of the easiest way to block inflammation, but maybe not the more, it's costly, so it won't apply to many people. I don't think that people with MDS will like to have like injected antibodies every 2 weeks or weeks. I don't know, but it's theoretically one of the most efficient way to block the inflammation in that specific context.
Okay. So if there aren't any more questions, thank you very much. A very, very interesting presentation. Obviously generated a lot of interest. I think there's a lot of people who want to work with you at the MUHC, so you're in the right place. Thanks everyone for your participation and for joining us today. And I wish you a great luck in your career. Thank you.